for hosting me today. And on behalf of the Arab School of Urology and the Arab Association of Urology, I would like to welcome you to attend my presentation. So I was assigned to discuss an important topic called sperm contribution to the developing embryo. First, I would like to inform that I don't have any disclosure to inform. My name is Dr. Hussein Kandil. I'm a fellowship-trained urologist. I'm practicing as a reproductive urologist at Faqih IVF in the UAE. So the topic today is about the contribution of the sperm towards the developing embryo. And what we have in hand right now is the basic semen analysis, which we all believe is not a sufficient tool for the proper evaluation of the functional aspects of the sperm. So by understanding the degree of contribution the sperm would contribute with in the development of the embryo, we would understand that we might need better tests and better evaluations to our male patients. So basically, the sperm contributes with so many functional aspects of the developing embryo, and hence the offsprings would rely on such contribution. The learning objective of today's talk is going to be summarized into three main topics. First, we need to understand the sperm contribution in terms of the parameters towards the developing embryo. Second would be the effect of sperm anomalies on the developing embryo. And finally, to know what are the conditions associated with productions of abnormal sperm, and hence abnormal contribution. So, understanding the sperm contributions to the embryo development. The sperm contributes by three crucial elements. Firstly, the paternal or the genetic traits of the father, which constitute the DNA and the chromatin. Secondly, and most importantly, the epigenome, which is going to be discussed further in detail. Secondly would be the oocyte activating factor, which induces activation of the oocyte and hence the fertilization process. Finally, the centrioles, which are responsible for the spindle fibers, which are also crucial in the early phases of embryonic development. Epigenetics or epigenome, which was first discussed in the 1970s, is something that is crucial nowadays. And when I was asked so many times by many of my colleagues that in the field of andrology, the pace of updates in terms of guidelines and recommendations is still on a slow pace. Now I can answer them, well, it's not that way. That's because we have so many new updates and new discoveries that I believe in the future and the soon to come future, it's going to change lots of our guidelines. The epigenome is one of that. And it's defined by the inheritance process of the phenotypic changes without the actual change in the DNA sequence. So it's really showing significantly in terms of the sperms or the gametes, especially when it comes to the male. So the sperm possesses a highly condensed nuclear structure within the nucleus, and they are wrapped around histones. These particles, they twist the chromatin around so that it's sort of protected. In one sense, it protects it, and in the other, it exposes it to the different forms of activation or deactivation, depending on the coiling of the chromatin. When it comes to the sperms, the protamine replaces histones, and that's the only place where protamine exists. And these protamines are the sort of a packaging system that is more tightly fitting the chromatin. And that's because at the end of the day, the sperm should have the chromatin very tightly compacted. And that's why chromatin is much smaller than histones, and therefore the packaging would be more fit. And after protamination, the residual histones actually becomes what we call nucleosomes, which also play a crucial role in the sperm epigenetics. So eventually, chromatin histones at approximately 90 to 95% are replaced by protamine by the end of the process. And therefore, the DNA is compacted. And it becomes less prone to have any external insults or any sort of hazardous changes to the DNA. It's sort of a protection. So let's just further elaborate. So we have the genome and we have the epigenome. The genome is the basic hardware that is not changeable. It's simply something that exists without any sort of change. And however, it is governed by a software. Let's consider it that way, which is called the epigenome. Epi means above, above the genome. So it does not change the genome itself. However, it sort of controls it like a maestro. Now, with certain exposures, there might be certain tagging with the epigenome on the genome, and therefore we have different forms of command, whether it's inactivation or suppression. What's interesting is that both the genome and the epigenome are both inherited. So consider that any kind of changes that happen resulting in an abnormal tagging using what we call the methyl or methylation of the DNA, which is sort of an epigenomic phenomenon, it will result in a change that would be inherited, and therefore we can see changes that are actually affected by such endeavor. So methyl group, which is the tag, we consider it as the switch of an on and off to the genome or the DNA. However, the histone modification, histones which I mentioned earlier, are simply like a knob that whether it turns it off slightly or on, but actually it just adjusts it. So in other words, epigenetics is nothing but like punctuating the same phrase with the same letters, which would yield into a different meaning at the end of the day. So both the sperm epigenetic transmission and the oocyte activating factor both would contribute in the early post-fertilization development, and therefore they would control the oocyte activations through the intracellular calcium homeostasis, stimulation of the oocyte to exit from the metaphase arrest, and initiation eventually to the early developmental processes. One of the contributions from the sperm is the centrioles, which plays an important role in the spindle fiber formation and the nucleation of the microtubules and basically the further development processes of the developing embryo. An additional epigenomic feature is the small non-coding RNA. Normally, a DNA would be transcribed into an RNA, and the RNA would translate into a protein synthesis pathway. The non-coding RNAs are basically an active sort of RNA that does not require any sort of activation to a protein synthesis. So they themselves are considered to be the active particles. These non-coding RNAs are abundant in the sperm, and they play a role in gene silencing, degradation, transcription, whether it's suppression and activation, and further regulation. One of the most common non-coding RNAs found in the sperm is the tsRNA, present in approximately 56%. And it plays a very crucial role in the sperm maturation and early embryo development. And hence, that would be part of our future studies to use it as marker for sperm quality and abnormal embryo development. The non-coding RNA, as I mentioned, they don't require any protein synthesis. They just act immediately and they activate the processes that would develop the zygote or the embryo after fertilization. There is sort of affinity or communication between the sperm and the oocyte. That's because there's a high affinity and specificity of the glucoconjugates of the zona pellucida in the ovum with the sperm. And therefore, there's an extensive crosstalks that happen and take place between the sperm and oocyte, resulting in the activation and further sperm decondensation now, because now we need to release the chromatin so it can undergo the replication process. We understand and we know today that the oocyte plays a major role in the repair of any DNA damages within the sperm. And we know that younger females repair sperms much better compared to the older ovum or egg. The second point would be the effects of sperm anomalies on the developing embryo. So we have an early paternal and late paternal effect. The early one would be failure to complete fertilization process, failure of fusion, the syngamy, failure of early cleavage. However, the late paternal effect would be implantation failure, miscarriage, or even developmental abnormalities. And this shows the significant contribution of the sperm towards the developing embryo. Thirdly, the conditions associated with the production of abnormal sperm contribution. So, trying to make my presentation more clinically oriented, we need to know what are the conditions that would predispose the male to offer a bad yield or a dysfunctional yield of sperm. So we have 6 issues to discuss today. Firstly would be the epigenetic changes. As I mentioned before, epigenome consists of DNA methylation or modification of the histones and the RNA disorder. DNA methylation. So methylation, we would consider it as simply a tag that would be attached to the DNA. And the more tags that would be attached, the more hypermethylation. In other words, this would suppress gene expression. And therefore, hypomethylation would promote gene expression. So now we can see how the epigenomic changes can change actual genomic function. Histone modification. So if there are certain chemical modifications that may take place within the histone, this would definitely alter the DNA binding ability. As we can see in the diagram, by the twist of the genetic material around the histone, areas would be concealed and others would be exposed, and therefore it's sort of an activation-deactivation process. And therefore, if an area was exposed, this would definitely favor the action of transcription factors and gene activation or even silencing. Sperm RNA disorders. Sperm carries thousands and thousands of different RNAs. What we are concerned about right now is the non-coding RNA type, which are involved in the modulation of gene expression, interfering with the mRNA translation through the different mechanisms. So, sperm RNAs are involved in transmission of the paternally acquired phenotype. And we know that sperm RNAs are capable of modulating early embryonic gene expression. And therefore, the general environment and environmental exposure, let's say, can affect the sperm RNAs and therefore can affect the phenotypic changes in the offspring. A study has shown that sperm RNA of obese males can explain the inheritance of obesity within the offspring of the obese patients. And now there's another example to see how epigenome can manifest clinically and through the process of inheritance in the offspring. DNA fragmentation and chromatin condensation disorders. So we know today that DNA has a robust power to predict fertilization in vitro. And it has been shown that the IVF outcome has significant difference between pregnant and non-pregnant groups based on the DNA integrity. So DNA by itself, in terms of its functionality and integrity, plays a crucial role. The impact of high DNA fragmentation involves actually, number one, poor day 3 embryo quality, low blastocyst formation rate and yield, and finally, low implantation and pregnancy rate. So sperm DNA damage could be used as a predicting factor towards the clinical pregnancy rate following IVF. The mechanism accounting for DNA fragmentation involves either inactivity of the endogenous nuclease, which actually is involved in repair, and therefore we end up with a dysfunctional DNAs with many defects and fragmentation, or abortive apoptosis activation, and therefore the cells get, apoptotic, therefore gets dysfunctional. And finally, oxidative stress, which results in a release of reactive oxygen radicals related from the mitochondria or sourced from the mitochondrial active phase during the epididymal passage of the sperms. So in other words, abnormal chromatin packaging and nuclear DNA damage appears to be linked. And there's a strong association between nuclear DNA damage and poor semen quality. One of the interesting phenomena is deficiencies in protamination. As I mentioned earlier, the protamine replaces histones to ensure a more compacted genome. So if we have deficiency in the protamine, then the chromosomes and the chromatin wrapped around the protamine would be exposed and therefore would be subjected to morphological defects in the head of the sperm. So the protamination actually involves 2 phases. phase 1 condensation, which is the phase where the histones are transformed into proteins, and this happens within the testes. However, phase 2, which happens when large disulfide cross-links appear, and this usually happens when the sperm moves from the epididymal head to the tail. And this, if passes smoothly, we can ascertain there is a chromatin integrity within the process. How about endocrinopathies? We also know that for a male who is presenting with infertility, whatever the the outcome, whatever the variables that we have to ensure an adequate and sound endocrine background. There is one interesting study that showed that patients presenting with high levels of reactive oxygen radicals and abnormal chromatin status with an associated hypogonadism, specifically low testosterone to estrogen ratio, when they were treated with letrozole, one of the popular aromatase inhibitors, things have improved. And we can see that through the tunnel assay, checking for the DNA fragmentation, that the levels went down significantly after treatment. In another study, it showed that IOI with sperm from infertile male patients treated with FSH led to 23% higher pregnancy rate due to reduced sperm DNA fragmentation rate. FSH by itself has been studied significantly in correlation with the DNA fragmentation, and recently there have been so many publications to show that FSH reduces DNA damage, anti-apoptotic effect, and induces maturation of the cytodia and germ cells, and increases the replacement of histones with protamine during the spermatogenesis. Varicocele, which is one of the most common clinical entity we come across as reproductive urologists. We also know that varicocele increases the oxidative stress, resulting in DNA fragmentation and lower spermatozoa fertilizing capacity. Interestingly, approximately 15% of patients with varicocele, even if they have normal semen analysis, they are rendered infertile. And therefore, varicocelectomy should reduce DNA fragmentation and improve sperm concentration, progressive motility, and morphology. Leukocytospermia. So, infertile men with leukocytospermia, they tend to have lower progressive sperm motility percentages, higher sperm DNA fragmentation indices, and high percentage of sperm with disomy XY and 18. And therefore, managing those patients would definitely, ahead of time, improve the outcome in terms of embryo development. DNA integrity is well correlated with peroxidase positive leukocytes. In one study, patients cured with leukocytospermia recorded higher pregnancy rates compared to those who had persistent conditions. Finally, the oxidative stress. So there is one interesting paper that's been published and recently with much fame that studied the oxidation reduction potential. And it showed that if we were able to measure the ORP levels, which shows the amount of oxidative stress exposing the spermatogenic environment, we can tell exactly what's going on, and this would definitely change the rates of what we call idiopathic male infertility. That's because lots of patients tend to have high levels of ORP. This paper showed us that the cutoff level of ORP is 1.34 millivolts per 1 million sperms per ml. And this would help differentiate between abnormal semen parameters in a high sensitivity and 40.6 specificity and 94.7 positive predictive value. ORP levels also help identify the altered functional status of the spermatozoa. So again, this is another entity that should be regarded prior to starting any ART treatment, since that many male patients tend to have high levels of oxidative stress. The redox dysregulation, which shows that increased numbers of defragmented DNAs, which has resulted from the increased exposure of oxidative stress, plays also a role in the poor development of the embryo. And idiopathic infertility, at the end of the day, would require or would benefit from a combined approach comprising oxidative stress analysis, sperm DNA fragmentation, and chromatin compaction analysis. In conclusion, Relying on the basic semen analysis seems insufficient to diagnose and manage male infertility. Spending enough time and effort aiming to improve seminal quality is crucial. Needless to say, lifestyle and behavioral factors play a role in the epigenetic abnormality and may contribute to abnormal embryo development. Finally, many available tools could guide clinicians toward the whereabouts involved in idiopathic male infertility, including the oxidative stress evaluation and DNA and chromatin assessment. These are my references. I'd like to thank you so much.